Hi, good morning, student. Today we are going to discuss the last topic, that is 21, and the title is Biotechnology and Genetic Modification. In this topic, we have to discuss about the genetic engineering and its useful applications. Now, firstly, we are going to discuss that what the genetic engineering is. Genetic engineering is the process of artificially altering genes in a cell to change the way it works. This could be to make the cell perform a desired function, such as making a specific protein, or to make the cell resistant to different factors. For example, some strawberries have been genetically modified to become resistant to frost by inserting a gene taken from cold water fish which makes antifreeze protein. In short, genetic engineering is the technique by which we take the gene from one organism and insert it into another organism to make the useful product for human. And in this technology, we use bacteria. So what are bacteria? We already have discussed it. Bacteria are useful to genetic engineering as they reproduce very rapidly, but still have the ability to produce complex molecules. Bacteria contain plasmids, which are the circular rings of DNA, into which new genes can be inserted, removed, or changed. There are also no ethical concerns about manipulating the DNA of bacteria, because if we are taking the uh, taking example as other organism, okay, or we just do some experiment on the animals, okay, so there are so many more ethical and moral issues are raised. But as you know, we are using the bacteria, so there is no ethical concern about the manipulating the DNA of bacteria. Now, bacteria can be manipulated to produce human protein such as insulin. You know very well that insulin is the hormones which is released from the pancreas. But some in some peoples, this insulin is not released in a enough quantity, due to which the person cannot maintain the sugar level. So for this person, we make the insulin from the genetic engineering technology. Now, what we do in this technology, the genes which codes for the desired protein is located and isolated using restriction enzyme. This isolated genes has sticky ends. The plasmid from the bacterial cell is cut with the same restriction enzyme. This leaves the complementary sticking ends to the isolated gene. The gene is inserted into the plasmid. The complementary sticky ends are joined using the enzyme DNA ligase. This forms a recombinant plasmid. This plasmid is inserted into the bacteria, which will then produce this protein as the inserted genes is expressed. The bacterial cell reproduced, making more cells which produce this protein. Now here you can see the diagrammatical expression of the same concept, that how the insulin is being produced for the diabetic patient. See, this is the cell of human, and this is a cell of bacterium. What we do, we take out the plasmid, that is the circular form of DNA, and cut it with the help of one enzyme. This enzyme called the endonuclease restriction enzyme, as you can see here. And we also take the chromosome of humans from the human cell, specifically from the pancreas. Then we cut the gene, or we isolate the gene from the human pancreatic cells, which is responsible to make protein. And now this gene is to be recombinant by the cut part of the plasmid. So here we can see that the recombinant DNA is made. This recombinant DNA is inserted into the bacterium. So this bacterium is called as recombinant bacterium. This bacterium is also called as a transgenic bacterium. Why? Because we are just changing the genetic of this bacterium. If the recombinant DNA is inserted into the plant, so it is called as the transgenic plant. Or if the same recombinant DNA is inserted into the into some animal, so it is called as the transgenic animals. Now see, in this diagram, the recombinant bacterium, when it's going to reproduce, okay, it's make its copy, because bacteria just reproduce very fast, so it's going to make its so many copies. After that, the insulin is to be extracted from these bacterium, and then extraction and purification of the human insulin. After that, it is filled in the bottle and launched in the market for the diabetic patient. So this is how the insulin is made, by using the genetic engineering technology. Now there are not only insulin, but also so many other products that we are making by using the genetic engineering technology. For example, see here, number 2, herbicides and insects resistant plant. Genes can be inserted into plants to make them resistant to the herbicides and insects. This means that less crops die, so farmers have a larger crop yields. Vitamin-rich plants. Some plants can be genetically modified to increase the number of vitamins in them. This is beneficial to places where certain vitamins are hard to find, the reduce vitamin deficiency. For example, golden rice is a type of rice that has been genetically modified to produce the beta carotene, which humans use to produce vitamin A. This reduces vitamin A deficiency in some areas. Now, what are the advantages of genetic engineering? So, loss of biodiversity. Okay, these are the disadvantages. Loss of biodiversity, potential development of weeds that are resistant to herbicides. Then GM crops are more expensive. GM crops may contaminate wild species by crossbreeding. Long-term health impact not known. Means if you are using this technology, so if there is any long-term impact that we do not know. Now, biotechnology. What is biotechnology? Biotechnology involves using microorganisms and biological substances to carry out functions to manufacture processes. For example, yeast is a microorganism which can respire anaerobically without oxygen to release carbon dioxide gas. This can be used in bread making. In the kitchen there is a yeast powder. This yeast powder, is added in the flour, and the yeast, when respire anaerobically, then it makes a high amount of CO2, which rises up the dough, and also it makes ethanol. This ethanol, is to be evaporated, okay, and the CO2 is used to rising up the dough. That's what, this yeast powder is used to make breads and to make pizza. The same yeast powder is to be used for the process of brewing. Brewing means alcoholic drinks. Because, when the yeast or the fungus respire anaerobically, it makes carbon dioxide gas and ethanol. This CO2 gives the fizzy nature to the drink, and ethanol gives the alcoholic nature to the drink. Now, this ethanol is also released during this reaction, which can be used to make biofuels that are used as an alternative to fossil fuel. Another use, you see, pectinate is an enzyme used in fruit juice production. Pectinase breaks down pectin, which is found in plant cells walls and is used to hold the cell wall together. Adding the pectinase therefore breaks down these walls to release the contents of the cell, which increases the yields of fruit juice. Now, it is also used in making the biological washing powder. As you know that yeast, secretes some enzyme. Yeast or bacteria use some enzyme. These enzyme break down the break down the uh, you know uh, the complex compound and turns the complex compound into the simple compounds. So just because of this, what happens, the strengths of starch, lipase, and fats or oil, okay, they are to be converted into the simple compound which can easily be washed out from simple water. The enzyme lactase can also be used to make lactose free milk. When lactase is added to milk, it breaks down the lactose into glucose and galactose, which can be safely consumed by lactose intolerant people. Because some people cannot digest the milk that contains the lactose sugar. So if the lactase enzyme break down the glucose into glucose and galactose. So this milk can easily be digested by the peoples who are lactose intolerant. Now, penicillin is a fungus used to produce penicillin. Penicillin, is the first antibiotic. The fungus is placed in the fermenter to keep it at optimum temperature and pH. So the penicillin yields is high. There is also an air inlet so that the aerobic respiration can take place. And all other microorganisms are killed to limit the contamination and competition. Now come to the diagram so that I could explain you very well. See here, this is the diagram of fermenter. As you can see in the diagram that the nutrients are introduced from the top of the fermenter. And now what are the nutrients means the glucose is there, amino acids are there, some minerals are there, okay. And from the another inlet, some microorganism are to be introduced. And when the fermenter works, then these microorganisms, specifically yeast and the bacteria, they increase their numbers and multiply, and they release high amount of enzymes, okay. And these enzymes are to be taken out. Now here, in the fermenter, how the temperature is to be maintained. For maintaining of the temperature, uh, cold air and cold water is to be run, okay. Now see the conditions. See here, aseptic precautions. Fermenter is cleaned by the steam to kill microscopic organism and prevent chemical contamination, which ensure only the desired microorganisms can grow. Secondly, the nutrients are needed for the use in respiration to release energy for growth and reproduction of the microscopic organism. As you know, when microscopic organism, especially yeast or bacteria, they respire, so they produce high amount of heat, okay, due to which the container temperature is increased. So for maintaining of the temperature is also very important. For this purpose, we just circulate the cold air or cold water around the fermenter. Optimum temperature is monitored using the props and maintained using a water jacket. This ensure an optimum environment for the enzymes to increase the enzyme activity and prevent denaturations. Now, what is the optimum pH? Bacteria and or fungus, they grow faster in a specific pH. So this pH is also be maintained. Okay, so pH is monitored using a prop to check it at the optimum value. For the microorganisms being grown, the pH can be adjusted using the acids and alkalis. Now, oxygenation, as you know that these organisms respire, so in respiration, they need oxygen. So oxygen is required for the aerobic respiration that has to be take place in the organisms which are inside the fermenter. Agitation steering paddles are used to ensure the temperature, pH, nutrients, and oxygen are distributed evenly throughout the fermenter. Now, the waste materials, see the waste materials which are released by these organisms, it is also be separated out. You know that outlet is there that you see here, exhaust outlet in the fermenter, which release carbon dioxide gas and other waste materials. So this is how the insulin is produced. This is how uh, the enzyme are produced which are used in the biological washing powder. So this is, this is the last topic of IGCSE biology. I hope you understand it very well. But still, if you have questions, so my WhatsApp number is there, my email address is there, and the same lecture is available in the my YouTube channel. The link is also available, and also the same lecture is on the TikTok. So if you have any question, okay, you can just write down in the comments or you can just WhatsApp me. Hope you understand it very well. Okay, so thank you very much. Bye.